My name's David Collingridge. I'm the Editor-in-Chief of the Lancet Oncology. I'd like to thank you all for coming. I'd also like to thank Johns Hopkins and Professor Jaffee for the opportunity to be in this room and be here this morning. So we're here today to see the launch of the Lancet Oncology's latest commission looking at future cancer control research priorities in the USA. And this commission was born out of the Moonshot initiative under the previous administration of Barack Obama. And also, it's based very heavily on the Blue Ribbon Panel and the Blue Ribbon Panel report that Professor Jaffee oversaw. That report came up with a number of recommendations across a wide spectrum in the oncology space. But there were some opportunities which weren't discussed in that report, particularly in areas around surgery, radiotherapy, imaging, and some other areas. This report goes into those areas in detail, but it also digs a little bit deeper into the Blue Ribbon Panel recommendations, puts some real granularity on what would you actually really spend money on, rather than big themes. What are the big buys that you could spend your research money on? So we have a great panel today who are going to run through the key highlights from the commission. We have a little change in the program. Dr. Lowry, the acting deputy director from the NCI, is actually going to speak first, because he has to dash a couple of blocks across town for another meeting. So without further ado, I'm going to introduce Dr. Lowry, and he's going to be talking about the Cancer Moonshot and the National Cancer Institute research. So.
Thanks very much. So, just so everyone knows, I'm from the government and I'm here to help you. So, let's just first start off about the Cancer Moonshot and what it was. It was intended to accelerate the pace of cancer research, but just in a subset of initiatives. It was not intended to represent all of cancer research. It was, the idea was to augment cancer research that we currently have. And it was focused on patients and on a limited number of areas of the portfolio that were poised for acceleration. In my brief remarks this morning, I would like to discuss two topics. The first is the rigorous process by which NCI has been implementing the recommendations of the Blue Ribbon Panel report. And the second is that the NCI is conducting a great deal of critically important cancer research that is beyond the BRP recommendations. And this is thanks to the strong bipartisan support for the regular appropriation from Congress that the NCI receives. But before discussing these topics, I would like to thank the authors of the Lancet Oncology report for building on the Blue Ribbon Panel report and its recommendations. And I would especially like to thank Dr. Elizabeth Jaffee and Dr. Chi Dang for their tireless efforts in the development and implementation of the Cancer Moonshot. Both of them were members of the Blue Ribbon Panel, as were many of the other authors of the Lancet Oncology report. And Drs. Jaffee and Dang are the first two authors of the report. And in addition, Dr. Jaffee is the chair of the National Cancer Advisory Board for the NCI, and Dr. Dang is the chair of the NCI Board of Scientific Advisors. Their leadership roles at the Ludwig Institute for Cancer Research by Dr. Dang and the American Association for Cancer Research by Dr. Jaffee just speak volumes about the respect with which they are held by their peers, the effectiveness that they have, and their commitment. So let me turn to the first topic, which really is the Cancer Moonshot. To jumpstart its implementation, new programs addressing 8 of the 10 Blue Ribbon Panel recommendations were initiated during this past fiscal year, despite the fact that the appropriation for the Cancer Moonshot did not occur until December, and the funding was actually not brought to NCI before the spring. And there are many additional programs that are slated to start in FY18, which will result in the launch of all 10 of the recommendations from the Blue Ribbon Panel. The funding announcements that led to the new research supported by NCI were vetted by the Board of Scientific Advisors, and every funding announcement clearly indicates that all applications should consider cancer disparities, which is an area that I emphasized during the 2.5 years that I was acting NCI director. I am pleased to tell everyone that Ned Sharpless has become the NCI director as of 2 weeks ago, and we are already working very closely together in my new role as deputy director of NCI. As part of the effort to accelerate progress with the Blue Ribbon Panel recommendations, all publications resulting from the Cancer Moonshot will be required to adhere to an open access publication policy and a policy of immediate data release upon publication. To promote and enhance data sharing, NCI last year established the Genomic Data Commons. And when the GDC was opened, it had 14,000 cases, mainly those from the Cancer Genome Atlas. But by forming partnerships with others, it has rapidly expanded both in size and clinical annotation and is expected to have more than 50,000 cases by the end of 2018. I'm particularly pleased to tell you that the AACR-sponsored GENIE database, which is from a number of NCI-designated cancer centers, is in the process of being de-identified with support from NCI and will become part of the GDC in the very near future. NCI has also developed new partnerships with other government agencies, the private sector, and the international research community to further the goals of the Moonshot in general and the BRP recommendations in particular. I'd like now to turn to the second topic, which is the equally important research that is not covered by the Moonshot. NCI is continuing to strongly support the many other areas of cancer research not encompassed by the PRP recommendations and the Moonshot. This includes training and mentoring the next generation of cancer researchers, supporting basic research, population studies, recently developed programs such as the Outstanding Investigator Award and the Research Specialist Award, the core grants for the NCI-designated cancer centers, the vast majority of our clinical trials, and most of our research on cancer causes, pathogenesis, prevention, screening, treatment, survivorship, and cancer health disparities. Progress towards understanding cancer, preventing it, screening for it, treating it, and maximizing further reductions in cancer incidence and mortality for our patients will only come from the combined efforts of the entire spectrum of cancer research, not from the Cancer Moonshot alone. So congratulations to all of you on this report, and I very much look forward to the remainder of the program and appreciate the invitation to get it started. Thank you.
Thank you very much, Doug. I'd now like to move into the report itself, and I'm delighted to introduce Professor Jaffee, who's the Deputy Director of the Johns Hopkins Sidney Kimmel Cancer Centre, who I'm sure you all know very, very well. Dr. Jaffee and Dr. Chi are the co-leads on this commission. So with that, I'll ask Liz to give us an overview of the research and clinical landscape in the USA.
Thank you, David. I wanted to thank you and your team for helping to put this together and coming up with even the concept. I think this is a very important concept. And as Doug had mentioned, and thank you, Doug, for coming today, he mentioned that this Blue Ribbon Panel report is really a roadmap for moving forward the priorities of the wonderful science that can be quickly translated into real treatments for patients. What this commission does is it takes, as David said, that those recommendations and tries to give more granularity to how to get there. So with that said, I'd like to start off by talking about the cancer burden research and the clinical landscape in the US, which is why we're even here today talking about moving forward with these priorities. Next slide. So, I mean, it's important to say that if you think about the world, cancer is really an epidemic. The incidence and death rates continue to increase. The number of new cases will increase by 54% by the year 2030, and the number of cancer-related deaths is expected to go up to 59%. So it's a real problem, and in the US, we're able to get treatments out to some patients. Outside of the US, when you look at the third world countries, it's a real issue. Next slide. So the death rates and the incidence aren't going up for most cancers. There are a few that are going up, but what is happening is that our population is aging, and so the number of total new cases and the number of total deaths are going up. So if you take a look at 2010, 1.5 million individuals developed cancer. This is expected to reach 1.9 million by 2020. Next slide. Same thing for the number of total cancer deaths. It was around 575,000 in 2010, and it's expected to reach 630,000 in the next couple of years. Next slide. The other major problem that we have in our country is health disparities, and this has to do with differences in biology that haven't been studied with research, as well as access to care. So in looking at this graph, although for all groups we're seeing again a decline in the total number of cancer deaths, you can see the red line is black males versus the black line is white males. Big difference in death rates for the same types of cancers. For women, it's not as bad, but it's still bad. And the blue line is black women, the green line is white females. So we really need to start putting our efforts at this. Next slide. Now, we know that the healthcare system has a true burden economically, and really the best way to deal with this would be to have more cures for cancer. So just to give you a sense of this burden, 1 in 12 Medicare fee-for-service dollars is spent a year on cancer care. That's a lot of money. 216 billion was spent in 2009, and that's expected to go up to over 458 billion by the year 2020. So this is, 2030. So this is really a significant increase, and it's a lot of our economic sector going toward healthcare. When you look at studies of even individual patients and their burden, you could see this is one study where 42% of the participants reported some significant financial burden. So for example, 19% were only partially filling their prescriptions due to a lack of funding. 20% took less than what they were prescribed. 24% stopped taking it entirely. So these patients not only couldn't afford it, but obviously their cancer wasn't being treated effectively. In addition, about 46% used their savings to help cover out-of-pocket expenses. And this is really sad. 46% reduced spending on food and also clothing. 68% cut back on leisure activities. So this is not only a financial burden, it's a quality of life burden. Next slide. So, and then cancer survivors. We need to do more work on cancer survivors. Cancer survivors are increasing, and that's great. That's terrific. However, they have problems. They have financial burdens. They have physical well-being issues, psychological well-being, social well-being, spiritual well-being. So, again, we haven't spent a lot of time on this before because this wasn't an issue, but this is now an issue. And again, one of the priorities from the Blue Ribbon Panel, as you'll hear from my colleagues, is that we need to think about survivorship. Next slide. So that's giving you a sense of where we are and why it's so important at this point to think about how do we do better. But also there's another reason, and that's that we're making amazing progress in basic science. So the investment by the government over the past 30 to 40 years has been significant, and we have come to a point where we're now hit a technological revolution. And this is really key because I think we forget that this took years and years of basic science, and we can't stop funding that basic science as well. So what is it that makes this a technological revolution? Well, new technologies have led to a better understanding of the biology of cancers, and this understanding is leading to not just new therapies, but precision-based therapies. And what does that mean? That means patient-specific based on what their cancer looks like, which means that hopefully it's going to be less toxic and more targeted, so better efficacy. Next slide. So just as an example, cancer genetics is one area where we've made amazing progress. And believe it or not, it was in 1977, quite a while ago, when, in fact, it was just soon after Nixon declared war on cancer, that the Sanger approach to sequencing came about. But it still took $2.8 billion and several decades to complete the Human Genome Project, which was a real advance in around 2000. So that was the first time we were able to sequence a whole genome. Wow. Well, look at how in a much shorter period of time we got to this next generation sequencing. Well, what does that mean? Well, that means today, not only does it cost less than $1,000 to sequence a patient's cancer, but we could take a small biopsy, just a small amount of material, and do the sequencing in a couple of weeks. So this has a lot of important information for us because not only do we learn about all of the mutations, and we now know that there's more than 4 million different genetic mutations in human cancers, but it also starts to tell us about the biology and also what pathways we need to develop new drugs against. Next slide. So the other area that is exploding, and I think all of you hear a lot about this in the news, is immunotherapy. And immunotherapy was the breakthrough of the year in 2013 in the journal Science. And so what is this? Well, what you're looking at here is you're looking at, and I'm sorry I don't have a pointer, but you're looking at a T cell. So it's the T cell that these immune agents are acting on, and those T cells naturally fight infection, particularly viral infection. Well, what's happening is T cells can also see these changes, these mutations in a cancer, and so they have the ability to kill the cancer. The problem is these T cells are regulated, and they have breaks. And so these new immune oncology agents release those breaks so the T cell can act as potent as possible. And that's what is happening. So why is this happening now? Well, we have the technologies and the knowledge from 40 years of studying regulation of T cells that we can now develop new therapies that can really have important implications for patients. So since 2011, when we had the first immunotherapy tested in patients, what we now have is over 25 FDA-approved immune checkpoints, immune oncology agents. These are all antibodies I'm talking about. Some of them are for malignant melanoma, and these are all metastatic cancer patients, so not patients with early disease, patients who have failed chemotherapy. But we're also impacting lung cancer. We haven't been able to do that in the past. We're impacting head and neck cancer, bladder cancer, kidney cancer. The other big advance is being able to engineer these T cells to specifically see proteins on a patient's cancer. And so CAR T cells, you've heard about that too. They've been approved now for 2 indications. CAR T cells are approved for acute B-cell leukemia in young people between the ages of 15 and 25, but also for a very aggressive B-cell lymphoma. So you could see in just such a short period of time, we've made this great progress, but it really took years of science, and it took this investment by the government. Next slide. So in the last few minutes, I just wanted to talk a little bit of how we're changing drug development in our country, and also I think in Europe and in the rest of the world. Cancer treatment is becoming personalized, as I keep saying. We're tailoring the treatment to a patient-specific cancer genetics and also their tumor microenvironment, their immune system. And so our old model, as you can see on the left side, you look at those patients. These are patients, let's say, with colon cancer, and the different colors means the different types of genes or mutations. We used to give the same treatment to all those patients, and so you'd have some people who would have some response, some who wouldn't, and then many who would have adverse events. What we're doing now is we've moved to taking the same group of people and saying, okay, let me take a little bit of your blood or let me get a piece of your tumor. What are the genes that we see in there? And then we target based on, so, if you have one set of genes, we give one drug. If you have another set, we give another drug. And we're seeing a much greater effect with less toxicity. Next slide. So there are two new models that we use for drug development. One is the umbrella trial, which I just described to you, where you have one type of cancer, but based on the genetics, you have multiple different treatments. The other is called a basket trial. And this is very interesting because we've recently had a very important advance as well in immunotherapy. And that's the basket trial is multiple types of cancer with the same genetic problem. And you give them the same treatment. Next slide. And this is an example. My colleagues, actually at Johns Hopkins, Young Lee and Luis Diaz, had reported only about 4 years ago that if you take colon cancer patients who have a mismatch repair deficiency, so their DNA, when it develops a mutation, they can't repair it. Well, what does that mean? That means that they make a lot of these mutations, and the immune system comes in and says, wow, it's like being a kid in a candy shop. Look at all these mutations I can attack. And so taking off the brakes with one of those immune oncology agents, you can now get a good response. So the blue lines are those patients with colon cancer mismatch repair who responded. The ones that did not have deficient mismatch repair did not. On the lower side is that basket trial approach where there are patients who have pancreatic cancer, gastric cancer, other cancers, who also had a mismatch repair deficiency, so the light blue and the black, and they responded as well. Well, why was this important? Several reasons. This is now an approved therapy, 3 years from first testing to approval. Have you ever seen that before at the FDA? That's amazing in my mind. And frankly, again, that was from the Moonshot where former Vice President Biden said we have to reorganize how the FDA works. We had rapid approval process, patients respond in first study, did not require a randomized phase 2 and 3 study, also important. Who wants to be randomized to something that's not going to work? And as I said, approval in 3 years. Next slide. So the overall goals, as you heard from Doug Lowy, is that the overall goals of the Cancer Moonshot were to accelerate progress in cancer, including prevention and screening, and this is taking the cutting-edge science and trying to get it to standard of care. We want to encourage greater cooperation and collaboration, and we also want to enhance data sharing. Next slide. So the Blue Ribbon Panel goals, and again, I want to thank the members here who are part of this, and also there are members in the audience who are part of this, was to identify major scientific opportunities that are poised to be accelerated by additional emphasis and funding, and to develop recommendations of opportunities that would be pursued through this Cancer Moonshot. And so the cancer research portion is named the Bo Biden Cancer Moonshot Initiative, and there's $1.8 billion that's been allocated over 7 years, and $300 million is already being used in FY17. Next slide. So and I took this from Doug and from some of the people at NCI. I hope that's okay, Doug. But I thought this was important to show this because you have to see what happens. So we can't do this without maintaining or actually increasing the standard budget of the NCI and the NIH, because this is extra money, not the money that's supporting the other research we need to keep getting the new discoveries. So what you see here is that in 2017 through 19, there will be an increase if we maintain the increase in the standard budget in what is spent. But the problem comes in that then the amount of money available decreases, and to maintain what we've already invested in, we're not going to be able to invest in anything more. So, this is a big advance, and this may be just my opinion. I think we still have to be fighting for more funding to make sure that the NCI and the NIH can continue to do what they're doing. Next slide. So I'm going to stop there and say it was a pleasure working on this Lancet Commission report. I'm truly committed to seeing that all of this comes to fruition. And the Lancet Commission report is really a roadmap for successful implementation of the Blue Ribbon recommendations. Thank you all.
Thank you very much, Liz. Our next speaker this morning is Professor Chi-Van Dang, who's the director of the Ludwig Cancer Research Institute in New York and a professor at the Wistar Institute. And Chi is going to be talking on the topic of selected highlights from the Commission, cancer research priorities in the USA.
Thank you very much, David. First, I want to thank David and Lancet Oncology for putting us to work. And you see the product today, which is quite significant. And I also want to thank Doug for his leadership at NCI and also for being part of the Blue Ribbon panel and hearing all the advances been made. And thank Dean Rothman and Johns Hopkins Medicine for sponsoring this event, and particularly all the authors for having contributed to this report. So as you already heard, this report distinguishes itself from the Blue Ribbon panel report in that it's an extension and an addition to the initial roadmap. So this provides an additional atlas of where we're going to navigate cancer research in the future. And so in the next slide, to put the report in context, I just want to illustrate here the domains of cancer care continuum from a report from the Institute of Medicine that spans across from prevention to screening, diagnosis, treatment, and survivorship. And if you look down the columns, you'll see that the many topics that are touched upon by this report, including tobacco control, diet, physical activity, chemo prevention, as well as immunization, which I'll touch on a little bit later. And then in terms of screening, there's also advances in technologies I'll also highlight. Then in the diagnosis column, you'll see that the usual ways of diagnosis are listed there, including the usual biopsy, but we also have part of the report where cancer imaging becomes very important in terms of advanced technology. Then in areas of treatment, I'll highlight further many parts of the report, in fact, highlights new treatment strategies and how the community could come together as teams to solve this problem. And then in the survivorship, particularly you heard from Liz already, in the US, there are over 15 million survivors that's existing right now. And then we'll touch a little bit on pediatric oncology, where many of the children who survive cancer, in fact, become survivors. And the next slide illustrates, again, across this continuum, the parts of the report that populates this entire spectrum. From the left, you can see immunologic prevention being parts of the report that fits into prevention. Then you move on into screening. We talk about early cancer detection and applying that to population science, not just the really hardcore science, but in the real world. How do you apply these? And I'll touch a little bit on that. And then if you look in the diagnosis area, there's a big challenge that was started by the Blue Ribbon panel report, which is creating atlases, which I'll describe a little bit more, that guide us in the future on why certain patients respond and other ones don't. And then in the same column, in terms of diagnosis, we have a whole section on nuclear medicine and advances in imaging, where you actually can couple imaging to therapeutics in new approaches. In the treatment column, there are many sections of the report that touches on this, including drug discovery. How do you expedite access to new drugs in clinical trials? And then including, which is not in the Blue Ribbon panel report, sections on surgical oncology, how surgical oncologists could participate in this team science approach to really reduce the burden of cancer. Radiation oncology, many advances, including new technologies and more precision radiation oncology, such as using protons and higher ion therapy. And then we also touch upon the revolution of cancer immunotherapy as well as precision oncology in that context. Then finally, in the areas of survivorship, there's a whole section about supportive care, and then pediatric oncology itself is quite unique, spanning all the way from prevention to survivorship. Then at the bottom, what you'll see, there's sections of the report that addresses topics that across the entire continuum from prevention to survivorship, including advanced technologies, and I'll highlight one of these, that can catalyze cancer breakthroughs from prevention, screening, and onward. You heard about health disparities from Doug and others, that this is a critically important issue that spans across the entire continuum from prevention to the end of life care. And then finally, big data and data sharing, while it sounds very simple, is another topic where the report actually provides some very practical approaches to solving this problem. To the next slide. I just want to highlight one of these sections, looking at advanced technologies to catalyze cancer breakthroughs. And this one, I want to highlight what is important about this report is not just areas of research, but it really is a wake-up call to the community to come together in team approach to solve very complicated cancer mysteries. So the key area is the convergence of expertise in engineering, computer science, and medical sciences to come together to solve these big problems, bringing new technologies as nanotechnology. And here I just want to highlight in the diagram on the right, it's just some work from Sangeeta Bhatia, who is a professor at MIT and also part of the MIT Ludwig Cancer Center there. And what she's done in her laboratory, she's quite creative in a sense of using nanosensors, that she's shown proof of concept that these nanosensors can be engineered to be injected into a host, and it can go about the entire body, circulate, and then when it finds the cancer, because the cancer environment has certain enzymes in it, that it flips on this light. And this light would then go into the urine that can be detected very easily. And she's been able to engineer this so that it's cost effective. And if it works, it could go into third world countries to really solve the problem globally. And that's just where cutting edge science can make a big difference in real world cancer problems. And then the other area, of course, is prevention and treatment. Again, circulating DNA mutations, some of it's pioneered at Johns Hopkins. Then finally, Liz touched upon how T cells can be engineered. And we just witnessed 2 FDA approvals for both leukemia and lymphoma. And I think this is another area that holds great promise, so research in the area of engineering T cells to go after not just liquid tumors, which is where I came from in my own discipline, but into solid tumors, which are much more challenging, and I'll touch upon this because the environment of solid tumors are quite different. Next slide. And then, of course, on the topic of big data and data sharing, the kumbaya of data sharing sounds very simple, but in fact, it is quite difficult because there are legacy systems. We also know that data quality really varies from institution to institution, all the way from clinical data to genomic data and onward. And so the report touches upon dealing with real-world issues, such as imperfect data. So the whole concept of embracing fuzzy data, albeit while it's imperfect, if you have enough large data sets that the signal that we're looking for would rise above the noise in the data sets. And this, of course, requires realistic data sharing framework. How do you, and Doug touched upon this, where the genome commons, the cancer genome commons actually is such an entity where public databases such as the GENIE program can be deposited. And of course, the report also encourages NCI to support more training of cancer data scientists, as well as NIH, because this is an area of great opportunity, and at least in my own opinion, this is where immigrants actually made a difference, and I'm an immigrant. And the other concept that coming out of this particular portion are data lakes, where individuals are able to deposit raw data, such as the one that the NCI is really catalyzing right now. It's going to be critically important, where people volunteer to put in raw data and then also be able to extract out and interrogate the data. Next slide. And then in the area of prevention, we're very excited about a whole area of immunologic prevention. And this comes from many years of lessons from smallpox, polio vaccines that we know have completely changed our world. Here, vaccines have been used against viruses that causes cancer, such as human papillomavirus that was pioneered by Doug Lowy, and I salute him for being recognized by the Lasker Award Committee and became an awardee of the Lasker. And so Doug was one of the pioneers of that. That made a huge difference in terms of cancer burden, not only in the US, but around the world, when HPV vaccine can be used effectively, and this is where implementation science also can make a difference. But really what the point of this is that we also learned from hepatitis V virus and C that causes liver cancer, and we see the reduction of liver cancer in Asia because of the effective use of hepatitis V vaccine. So from these lessons, we're very encouraged that cancers, with the alteration of the genome, and this is what you heard from Liz, it's a genetic disease of somatic mutations. In essence, cancers are infected with altered genomes. That's a way to look at the cancers. We are infected with an enemy from within where the DNA sequence has been broken and the brakes are released, and then the accelerator on for the cancer cells to grow. But in doing so, it makes a lot of mistake, and it presents some of these alterations and appear as if it's infected only from the inside. So there's a great hope that we will discover what we call neoantigens. These are antigens that are produced by the cancers that are recognized by our immune system as being foreign. But as you heard, that there are breaks, that now there are drugs to uncover those breaks. But the promise here is how do we use various approaches for a cancer vaccine, such as using DNA sequences, which I'll get back, peptides, which are what's been used for many years, and then also there's been approval for dendritic cell vaccines as using the tumor cells themselves. Now, with regard to DNA sequences, this is the technology to keep an eye on, because instead of making peptides to immunize, such as attenuated virus for viral vaccines, what the DNA sequence has been able to do, and this is where research have allowed us to advance, is that you can encode the entire protein or peptide within the DNA sequence, and every piece of DNA have the same quality or safety profile. And this is work done, pioneered at many places, including at the Wistar Institute by David Weiner, where you can engineer DNA sequences to really produce vaccines against various diseases. More recently, a publication on the Zika virus vaccine that was developed by David and just published in the New England Journal. So this is an entire area where I'm very encouraged by, because this is where personalized vaccines can come into play, where individual tumors from patients can be sequenced, neoantigens can be predicted by computer scientists, and then the vaccines can be made purely by synthesizing a DNA that makes the vaccines that can be injected right into the muscle, just as you would get vaccines for other viruses. Next slide. And then in the area of diagnosis, again, we salute the Blue Ribbon Panel report, so we're saluting ourselves, really, on the Cancer Atlas, and we want to extend this out, and I just want to spend a moment here to describe what we mean by this. So the cancer cells don't just exist by themselves, particularly in solid tumors. The diagram there just to illustrate, in the lighter circles are the cancer cells, and then you have the other colored cells are in there are immune cells, and the green cells are the fibroblasts. So the mixture of cells that are in a solid tumor is quite complicated, with blood vessels feeding it. And so what we don't know at the moment is when we give chemotherapy or immunologic therapy, what happens to all these cells? Which goes away, which remains and then causes problems for cancer patients when it relapses. And so only way we can understand this is to be able to collect tissues, but do it in an intelligent way to connect them with clinical trials that are informative, so we can follow the clinical trial along and know what has altered in the tumors that causes certain tumors to completely go away and others relapse. And this allows us to really sample tumors over time, coupled with clinical trials, and also to use advanced technologies where now we can study single cells. You can tease out from the tumors individual cells, and then with the latest technology to look inside those cells, what is instructing them in terms of the mRNAs. This is called RNA sequencing at a single cell level, and then to be able to reconstruct all these data. And again, this is another massive amount of research that's going on that's going to generate a lot of data that needs data collection, computer scientists to integrate this, to really allow us to create atlases that allows you to look at the three-dimensional composition of the tumor as well as over time, so not only in space but in time within various clinical interventions. And this is how we're going to change how we're going to take care of cancer. Next slide. And then I just want to really spend a particular moment on pediatric oncology, which is very special. It's also a section in the Blue Ribbon Panel report. But we extend this and really underscore some of the importance in this area, because at least from my perspective, a life saved of a young child is a world saved for many years. And I've seen this in a number of patients. But pediatric cancers are very different because they, by nature, are different. There are rare diseases where the genes are swapped to produce chimeric proteins that we know exist for many years now, but the technologies have not allowed us to attack those very specific alterations only in the cancer cells, not found in normal cells, such as chronic myelogenous leukemia, where we have experienced Gleevec that went after one of these chimeric proteins. So this is an area where the Blue Ribbon Panel report really underscored the importance of finding drugs against those fusion proteins, albeit these were rare diseases. And then another area is the focus on adolescent cancers, and also because of the spectrum of when we cure these kids, that they're going to be growing up to adults, that there's also issues of ethnic disparity, both at the biological levels and as well socioeconomic level that needs to be addressed. And here we can learn from the pediatric oncology community on how research networks are created, because no single entity can really collect enough patients to really understand. When we collect all the data together and collaborate, you can really understand these diseases. And then the pediatric oncology also span across into survivorship, as I already mentioned before, because more than 80% of children survive cancer for more than 5 years. And again, with all the complications you heard from Liz already, that has to be contended. So the report also addressed the issues of various issues around survivorship. Next slide. So then the final slide here is really just to emphasize, and you'll hear more about this by colleagues following me, a call to action. And then the way we're looking at this is that it's not all about resource. It is in part about resources, you heard from Liz, but really it's a call to action and convergence of not just resources from various stakeholders, but convergence of will. The convergence of will to defeat a problem that is going to continue to face us. And this allows us to really challenge all the funding agencies, as well as policy-making agencies, companies, governments, universities, patient advocacy foundations, as well as cancer research organizations, to come together through alignment, collaboration, transparency, and this is where data sharing is going to come in. And finally, most importantly, I think if we're going to make a difference globally, is affordable cancer health. And thank you very much for your attention.
Thank you very much, Chi. Our next speaker this morning is Professor Scott Lippman from the University of California, San Diego. And Scott is going to be talking on the topic of cancer prevention, detection, and health disparities. Scott.
Great. Thank you, David, and Liz and Chi for this wonderful opportunity to participate in such a timely and important session. So I'll talk about some fairly diverse topics: cancer prevention, detection, and health disparities. And sometimes it's a problem to be such good friends with who the organizers are. When I sent my slides in a couple days ago, Liz Jaffee said, can you just tell one story? I think not so easy with this, but I did try. You can let me know. I made some changes. And I also want to say that I don't know if I should say speaking on behalf of, maybe you can decide if that's if you want to be in that same company, but certainly Dean Ornish and I worked very closely on this, so I hope I got it close. So next slide. So here's the sort of story or theme when I started thinking about it, and that is the fact that technology is transforming much of what we do in prevention and disparities research, and in different ways, and I'll show you how. I'll talk about geographic information systems, very important, disparities and so on, physical activity and sedentary behavior, how we monitor these things with a lot of technology. In the past, it's been very subjective. Now we're talking about very quantitative ways. I'll talk a little about circadian rhythm effects, big data, the pre-cancer atlas, and precision prevention. I was excited that I was fortunate to co-chair the Moonshot Pre-Cancer Atlas with Avi Spira. Liz was there and some others, and the RFA just came out recently, and I was very excited to see that there were 3 RFAs on a prevention PCA and 3 on therapy. That's really exciting, because a lot of the prevention work doesn't necessarily gain the same sort of enthusiasm as therapy. But those of us who have been doing prevention for 35 years look at it differently. Next slide. So, I'm going to use where I live to illustrate some of the points that I think in the beginning cut across a lot of areas. So This is a type of geographic information system. It's out there, people have it, but this is, it's a different way to look at your region. So San Diego County is a very large area, 3.3 million residents, the fifth most populous county in the US. In San Diego, Hispanic populations outnumber the rest of the US by about 2 to 1, and the same for Asian and Pacific Islanders. And you can see the points, but the point is it's not homogeneous across the region. There are definitely areas that are very high in Asian Pacific Islanders and distinct areas that are very high in Hispanics. And this is extremely important because if you want to develop some sort of targeted intervention, screening or other things, it's important to know where to direct your efforts. And I'll talk a little about that. Also, just of note that San Diego County has about 43% non-English speakers. So I'll talk about a disparity involving them. It's 21% in the US, and you can see the poverty data. Next slide. So a major problem in San Diego and in the US is the dramatic increases in liver cancer and its precursor lesion, which is NASH. This tracks, it's much more common. It's in Hispanic people. It's the second most common cause of cancer death in the United States in Hispanics. NASH is highest in U.S. Hispanics, and this is related to obesity. So on the map on the left there, you can actually mark where these patients with liver cancer death are. So if you wanted to try interventions or prevention or so on. And then, we need to understand better what drives NASH to become cancer. That's obviously a really important question, and there's work in several different areas going on. I just show this bottom one from Karen Nelson at the J. Craig Venture Institute. She's the president there, and she was the first to sequence the human microbiome. But in any case, this is just to show you an example of where her data supported or found evidence of certain bacteria that seem to promote fibrosis in NASH. And in that upper right-hand graph with the bar going up is that NASH is a major predictor, fibrosis in NASH is a major predictor of liver cancer mortality. Next slide. So we don't often think of this as a disparity, but it certainly is, independent of anything else, and that is the fact that if people can't speak the language that you're trying to talk to them about various screening or other behaviors, it's obviously very ineffective. So, if you go to a certain high area for colorectal cancer and you try to talk to people about getting screened, it's just not going to work. And so what we and others have done in the case of people that speak Hispanics that don't speak English is to hire promotores that are not health specialists, but they speak the language and can convey the ideas to people. And these have been very effective at promoting screening, for instance, in Hispanics and in colorectal and breast. By the way, on the right, you can see that of the 43% non-English speaking, most are Spanish, which is what you would expect, but there's a lot of Asian immigrants as well. Next slide. And then the prevention is just shown here. We have very active quit line that we monitor nationally, and and I just want to point out that here it's been very effective. And one of the things it did is it actually addressed the issue of people who are Asian who don't speak English. And they showed in a randomized trial published in JNCI a few years ago that when they had a line that had 3 Asian languages that was so when people called in, they could help them. It was very effective. Next slide. A lot of you have heard of this phrase, and that is that sitting is the new smoking. You can see the evolution of man from walking, hunting, to sitting in front of a computer, there are about 15 different cancers that are increased in relationship to sitting versus physical activity. So this is turning out to be a very important factor and probably overrides physical activity in many settings. Next slide. I just want to show in terms of, again, the technology that on the upper right, you can monitor individuals that are wearing various devices, and you can see how much they move during the day and quantitate this. You can quantitate disparities by access to healthcare on the bottom right, and you find that people that live far away from hospitals don't often get the same care. And then you can also map obesity as well, which is an important risk factor for a number of cancers. So another kind of technology. Next slide. The study of the built environment and activity, some of you may not have heard of this before. I hadn't until recently, a few years ago, and that is the fact that the environment, the cities you live in, can directly affect cancer and other rates. So that there are cities, as you can see on the left, designed for cars, good freeways and so on, and then there are cities designed to move people, where you can walk. There's crosswalks and there's sidewalks and so on. And what this study showed, which was published in the Lancet in 2016, not Lancet Oncology, but your partner, and the study showed that neighborhoods like on the right, which are walkable, and it was done actually internationally, picking certain cities that were diverse in this issue, showed that active supportive neighborhoods where activity was about 90 minutes more per week, so not a lot per week, than least supportive. And there was an editorial with this that suggested that 2 million lives could be saved annually worldwide. Not all cancer, but certainly cancer is part of it. And so what this has done now is that globally, nationally, and locally, people that are involved with developing transportation, cities, and landscapes, and so on, are taking this very seriously with the help of CDC and World Health Organization and others. Next slide. So you are when you eat. This is a growing topic related probably to circadian rhythms, but just to warn you, that you see the two mice there, the one is a red box and one is a green. If you look on the far left, you'll see that in the red box, these mice are exposed to food 24 hours a day. It's available to them. And then the 8-hour mice only have food available for 8 hours, and they can eat as much or little as they want. The issue is the duration time. And so what you see in the far left is that the red line is dampened compared to the blue line, which is a more typical circadian rhythm. And that correlated with an increase of breast cancer in the red, mice that were eating around the clock. Next slide. And this is now moving into humans. It's an interesting study. You can see at the top there's an attempt to, in the fasting eating, to show that the restricted eating box is, doesn't eat for about 13 hours. I mean, there's nothing magic about that, but for a period of time versus unrestricted eating. And the difference is that cancer is reduced. Cancer recurrence is reduced by 40% in breast cancer survivors who have fast for 13 hours or greater. And that circle in the middle is interesting because each ring of the circle is a person. And what you can see is that, and you can see the midnight, the sort of the hours of the day. And you don't find, I mean, you see that there are a lot of people that are snacking a lot and throughout the day, and that could be bad. Next slide. So, when you eat is important. This is an example of big data doing things that are just almost unimaginable, starting to do this. And this is a study from Hannah Carter that looked at the germline and in a very complicated way, and was able to show that there are ways to determine ahead of time where a cancer will develop and when it will develop. Where and how. And this is just a cartoon. This got a lot of attention, and you can just see the areas of the cancer above the line and below the line. The how is, which mutations are involved. I sort of refer to this as the Minority Report. If those of you know the movie, what this could mean for prevention and screening if you knew, what the key areas were to focus on. Next slide. Atlases. I borrowed your circular thing, Chi, at the upper right. For the atlas, but this is a big deal in prevention. I mean, Liz and Chi have talked about this. Maybe I can be a little more blunt by saying that in terms of premalignancy, it's been a complete black box. I mean, we know very little. It's hard to find a cancer now that we don't classify by certain subtypes, molecular subtypes, and signaling pathways and treat that way. Histology really is back in there, just starting to change of, histology, dysplasia, whatever, and it's, so it's hard to do precision approaches in that kind of setting. And in fact, in the 35 years I've been doing this, I probably have published more negative studies than most people. Maybe it could be the record. I don't know, but it's hard when you're shooting in the dark. And so, to me, we have to regroup and focus on these atlases. And you can see in the middle there how complicated they are. I mean, you start to see changes in microenvironment occurring, no question about this, in the premalignant state and effects of the germline and so on. But then on top of this, the immune atlas, which is antigenicity and so on, and which cells, and M1 polarity and things of this sort. And so both are important, and it's incredibly complicated. We found out at this meeting that we were at that, like, mast cells, for instance, are important as well. And it's not only whether mast cells are there, but it's actually the distance of the mast cell to the neoplastic cell can have a huge effect. So, I think it really is complicated, and in a way reminds me of a quote that I've always liked from Bert Vogelstein when these kind of things come up, saying that we know prevention is complicated for a lot of reasons, but that doesn't mean we shouldn't do it because we have the tools now, and the impact is phenomenal. And Chi, I know either Chi or Liz commented on the HPV vaccine. Can you imagine if we develop a vaccine towards these? You don't have the same urgency, that's what I'm right there, but I'll talk, develop a vaccine that 1, 2, or 3 injections will be lifetime protection. I think it's only one, right, for the HPV, maybe. But in any case, this would change everything. So I think it's worth the effort. Not sure it'll finish in my lifetime, but it's definitely worth the effort. Next slide. Just go through this quickly. This was reviewed earlier, but this is Lynch syndrome, which is characterized by, I think Liz mentioned, mismatch repair defects or microsatellite instability. It turns out microsatellite instability is extremely common in about 30, maybe, I don't know, was it 20 cancers or something, are microsatellite unstable. But it started with Lynch syndrome, and this I think is a paper that she mentioned. But if you look at colon cancer, for instance, you see a subset on the left there that have very high mutation rates. And those tend to be MSI positive hypermutators compared to the rest of colon cancer. And those patients with the very high levels of mutations respond extremely well to checkpoint inhibitor for therapy. Next slide. Well, it turns out that this is translating to prevention. And I think the Lynch syndrome was a major demonstration project, I believe. And it's not even for this reason. There are other aspects of what you do with colon cancer in the patient, because Lynch syndrome, of course, develop other cancers. But what's amazing is that if you look at the man on the right there on the bottom, who they know has Lynch syndrome, but has been screened extensively, is in the context of a very intensive study, and has no cancer anywhere, no polyps, nothing, and yet still has T cells to FSP, So FSP-specific T cells, as you can see, going to HNSPC, which is the same as Lynch syndrome. So what that means is, at the earliest stages, in healthy people with Lynch syndrome, you're seeing evidence of immunosurveillance, which is really important when you start thinking about vaccines and other things to have that. So it's, I think, a real opportunity. Next slide. BRCA1/2, people know about this, certainly in the context of breast, and got a lot of attention with Angelina Jolie. Next slide. So if you want to talk about precision prevention, I can think of no better example than this, and where the idea, again, that Liz commented Shay about knowing the science. What we know about breast cancer and BRCA1 positive carriers is that it's ER negative, triple negative, aggressive. That's what we know. But there's been a few things in literature that haven't been so consistent with that. Anyway, this is a paper published a year ago from Nolan from Australia, which then, now that we have the tools to work with very small specimens and things that have made it challenging for pre-malignancy versus cancer, that you see in the left there, that it turns out that the earliest progenitors of BRCA1 mutant cancer are in fact hormone responsive. No question about it. So you can see here that rank L is one of the molecules that's a key regulator of these progenitors. And if you inhibit it, it prevents cancer in mice. So I think this is true. I'm not 100% sure, but I'm pretty sure that this is already begun or soon to begin an international trial of a rank L inhibitor in BRCA1 carriers. And it's the other convenient thing about this, of course, is that denosumab is a drug that inhibits rank L and is FDA approved, and approved globally, I think, for preventing osteoporosis. So the safety profile is extremely well known. So it's just all set up. So I think that this is a really exciting study, and we'll hear more about this in the future. Next slide. Let's finish with two slides on pancreas. Liz told me to cut the other ones out, so that's a good thing. And the main reason to do this is, again, just so you know that when I use the term interception, we're referring to a type of cancer prevention, active. Liz Blackburn coined that term. But in any case, in pancreatic cancer, which is arguably the most severe and devastating cancer there is, annual screening with MRI or ultrasound, endoscopic ultrasound-based approaches has actually, for the first time, shown what appears to be a benefit, and that's the Vassan paper. This is the first signal, the first real signal that screening may be beneficial in this disease. And we know how well treatment works in pancreas cancer. So I think anything like this is important. And then you can screen for familial syndromes. The germline risk, it depends on the setting and clinic versus whatever, but can be in the order of 15% of pancreatic cancer have a germline mutation, dominant germline mutation. And interestingly enough, coming back to the BRCA1, by far, 2/3 of patients with pancreatic cancer, 2/3 of the 15% are actually have BRCA1/2 mutations. In fact, we all know about how common it is in breast and ovary cancer to screen. The third most common is pancreatic cancer, and there are certain settings where guidelines do. Doug, did you want to make any final comments? Or no. I'm giving a talk up at NIH. Sorry. It wasn't something I said, right? No. Okay, good. Okay, good. I saw you at the edge of the chair. That was fun. Anyway, so BRCA1/2, a lot of you probably don't know. I didn't know until I got involved, until I ended up on the AACR board sitting next to Liz, and I learned a lot. But the fact is that it's a really important issue, and in different high risks. And then on the bottom, you can see this is just earlier late stage pre-malignancy, and those arrows are pointing to immune infiltrates, showing evidence of immune surveillance in patients with pancreatic premalignancy. Next slide. Yeah, I think this is the last one. I just want to show that there's a lot of interest in immune prevention in pancreas, and a lot of that's driven by Liz Jaffee's classic work shown here and other papers. But in this aggressive mouse model, a KPC model, she found that a KRAS vaccine with Treg depletion did improve survival. That's where the arrow is, but only when given early. And this is, it may not look like much, but this is in a very aggressive model. So this is a really important signal. And again, to move it earlier, Steve Schoenberger and Craig Venter and others are taking a different approach, so be complementary, look to develop personalized vaccine strategies, thinking that the number of neoantigens will be lower, and you may be able to develop a personalized approach. So a couple different approaches that are going on, and again, as Chi mentioned, in the prevention setting, you do have, you don't have the same ticking time bomb. You do have, even though it takes a little longer, you could make these personalized vaccines where you can actually test every neoantigen immunologically. And next slide, I think it's it. Oh, so call to action. So what I'd like to say is that regarding cancer prevention or interception, the time is now. From lifestyle to immune interception, I hope I've convinced you that there's tremendous promise. We have the tools, we have the technology, we have the will, as was referred to earlier, to prevent cancer. And although it'll take time and a lot of work, this would be make an incredible public health impact for the better. Thank you.